Hello everyone, welcome back to the session. So we are discussing about the cell organelles under the chapter cell biology. Okay, so we are discussing about the in yesterday's class we have discussed about the peroxisome. Okay, an important organelle. In today's class we are going to discuss about the ribosomes. Okay, we'll discuss it. See, how can we define the ribosome? So we have discussed about the endomembrane system. So whenever we are start to study about the organelle, it is mandatory to study about the membrane bound, whether it is a membrane bound or not a membrane bound. If it is a membrane bound, whether it is made up of single membrane bound or double membrane bound organelle. Okay, and what is mean by endomembrane system? What are all the organelles which is coming under the endomembrane system? Which are all the organelles or not? Okay, all these are an important thing. Keep on remind you the same thing again. Why? Because it is important. Okay, now how can we define this ribosomes? The first thing will be it is non-membranous. Just to make a note of it, it is non-membranous ribonucleoprotein particles composed of rRNA plus protein. You should know ribosome is equal to rRNA plus protein is equal to ribosome, right? Now, see this ribosome and protein responsible for protein synthesis. So, this ribosome is mandatory to synthesize a protein. Okay, so protein synthesis is technically called as a translation, which is present in all the cells. Okay, see ribosomes are non-membranous ribonucleoprotein particles and that contains rRNA plus protein, and this ribosome is very important for the synthesis of proteins. Okay, this is present in the living cells. Okay, so you should know ribosomes are the site of the protein synthesis. This can be expected in the question. Okay, now see what will be the discovery and the history. It will be discovered by George Emil Palade. He is the one discovered the ribosome under the electron microscopy, and he named this George Emil Palade named the organelle called the Palade particles. Why? Because he discovered the ribosome. That is why he named ribosome as Palade particles. Okay, and he was awarded Nobel Prize for the work on all the cell organelles. Okay, and you should know bioplast. Bioplast is also called as a mitochondria. Before the term mitochondria exists, some scientists have named mitochondria as bioplast. After that, Benda came, replaced the word bioplast, and he named the word mitochondria. So, like in the same way, here Palade has named the ribosome as a ribosome Palade particles. Okay, just all these things are having a connection. Now see here, ribosomes are found in. See here, ribosomes are found in the prokaryotes, free in the cytoplasm. Okay, see here, this ribosome can exist in two forms. Okay, one, either it will be membrane bound or it will be free. Okay, it is not bound to any of the membrane bound organelles. Okay, so it is not present in any of the membrane of the organelles, or it will be free. Okay, so it can be present in the organelles membrane, or it can be free in the cytoplasm. Okay, so that is what this ribosomes are found in the prokaryotes, which is free in the cytoplasm, and in the eukaryotes. So in eukaryotes it is free in the cytosol, bound to rough endoplasmic reticulum. Okay, it is bound to rough endoplasmic reticulum. Outer nuclear membrane, it is also present in the mitochondria and the chloroplast. Okay, so in prokaryotes the ribosomes are free in the cytoplasm. It is not membrane bound in any of the organelles, because there will be no membrane bound organelle seen in the prokaryote. That's the reason. In eukaryotes, it is also present in the, it is free in the cytoplasm. Okay, it is roaming free in the cytoplasm, and also it is bound to the rough endoplasmic reticulum. So this ribosome we can call them as a membrane bound ribosomes. Okay, it is bound to the rough endoplasmic reticulum and in the outer nuclear membrane. Okay, so it is also present in the nucleus. Why? Because nuclear DNA is also there. Okay, so outer nuclear membrane and outer nuclear membrane is the one continuous structure is a rough endoplasmic reticulum, no doubt in that. And the ribosome is also present in the chloroplast and the mitochondria, and the ribosome which is present in the mitochondria and the chloroplast will be the 70S ribosomes. Okay, now See, this is what a structure. So this is the nucleus. Okay, so this is the nucleus and we are having a nuclear envelope. So in the nuclear envelope, that is outer membrane and the inner membrane together, we are calling them as a nuclear envelope. Okay, so in the nuclear envelope, they are having many pores. We are calling them as the nuclear pore. Okay, so this the endoplasmic reticulum with the ribosome, we are calling them as a rough endoplasmic reticulum. Okay, so and the rough endoplasmic reticulum structure is cisternae. Okay, cisternal endoplasmic reticulum, and the ribosome will be embedded in the membrane. So this is what a ribosome, and this is a membrane bound ribosome, and we are having no ribosome. That endoplasmic reticulum is called as a smooth endoplasmic reticulum. All these things have been discussed in the previous class itself. Okay, now what are all the chemical composition of the ribonucleoprotein complexes? See here, ribosomes are ribonucleoprotein complexes. So it is the mixture of rRNA plus protein. So we are ribonucleoprotein is nothing but a RNA protein. Okay, ribosomes are ribonucleoprotein complexes. Okay, so what are all the components of the ribosomes? Okay, we are having rRNA which is present in the ribosome. Okay, see, ribosome is nothing but which is a combination of this rRNA and protein. Okay, together we are calling them as a ribosomes. Okay, which is essential for protein synthesis. See here, rRNA, ribosomal RNA. Okay, ribosomal RNA, 60 to 65%. This is the structural and catalytic role. So this RNA is responsible for structural and catalytic role. Okay, so this is very important. So in ribosome, 60 to 65% we are having rRNA. Okay, and proteins, 35 to 40% of proteins, it is the structural stability. Okay, now this rRNA is acting as a ribosome. That ribosome is nothing but a peptidyl transferase activity. See here, this is having the catalytic role. Catalytic, who is going to catalyze the reaction? Enzymes, and this rRNA is acting as an enzyme itself. Okay, so that enzyme, that ribozyme. Okay, so ribozyme, that is called as the peptidyl transferase activity, and this enzyme is very, very important for protein synthesis, that is in the translation process. Say in the replication, DNA polymerase is important, in the transcription, RNA polymerase is important, and in the the transcription, RNA polymerase, translation, peptidyl transferase activity is very important. We are going to see one by one. Okay, so see here, as of now, just know that this ribosome, which is nothing but rRNA plus protein, simply we can call them as a ribonucleoprotein complexes. So mostly 60 to 65% it is made up of rRNA, and 35 to 40% will be the protein, and this rRNA is for the structural and catalytic role of the ribosome, and this protein will be the stability of the structure. Okay, so this is what. See this, look at this picture. So this is what a ribosome. Okay, and this is mRNA. Okay, so this is mRNA, and this larger part is called as the larger subunit. So whenever we are studying about the prokaryotic and eukaryotic differences, we have studied about the 70S ribosome and 80S ribosome. Okay, so the 70S ribosome can be divided into two subunits. One will be 50S and another one will be the 30S. Okay, so this will be the larger unit and this will be the smaller unit. This is how the structure will be, right? Now see here, this is a ribosome, and there will be an mRNA. Okay, and this is a larger subunit and this is a smaller unit. This two subunit, we are called them as ribosomes together. And see, with the help of this ribosome, protein synthesis will be taking place, addition of the amino acids. Okay, so this is what involving in the protein synthesis. See, all this replication, transcription, translation, you are going to see in the molecular biology chapter. We are going to discuss about that. Okay, now, so what are all the ribosomal types? So as I have mentioned you before, we are having two type of ribosome. One is 70S ribosome and 80S ribosome. You definitely have to know what type of ribosome seen in which of the organelles. Okay, see here, in case of prokaryotes, 70S ribosome will be there. In case of eukaryotes, 80s ribosome will be there. Okay. So in eukaryote, where exactly the ribosome will be present? In the cytoplasm. So in the cytoplasm, we are having 80s ribosome will be there. Okay. And so in the ribosome which is present in the prokaryote will be the 70s in prokaryotes, and also where in eukaryotes which organelles they are having 70s ribosome? Mitochondria and the chloroplast. Okay, see here, prokaryotic organisms, all these organisms are having 70s ribosome. Besides, mitochondria and chloroplast, they are also having the 70s type of ribosomes. Okay, and in eukaryotic cytoplasm we have seen this 80s type of ribosome. Okay, and the subunits of this 70s ribosome will be 50 plus 30, and the subunit of 80s will be 60s and the 40s, and I have explained you why this has been divided into larger and smaller subunit, what is meant by S, all these things. Okay, Swedberg unit is yes, is not additive. See here, Swedberg unit, it is a Swedberg unit. S stands for the Swedberg unit. Okay, this is how the structure will be. Okay, see, this is a smaller subunit and this is the larger subunit. This is how both the subunit are holding together, right? Now, so we are going to discuss about now a prokaryotic ribosome. Prokaryotic ribosome, just now we have studied the 70s type of ribosome will be seen in the prokaryotes, and we are having two subunits in the 70s, one is 50s and 30s. We know that 50s ribosome is considered as a larger subunit and the 30s subunit is considered as a smaller subunit. Okay, so in the 50s subunit, again this is also contains an rRNA and protein. Here in smaller subunit also they are having the rRNA plus protein. Okay, so in the rRNA of this larger subunit, again this 50s has been divided into 23 plus 5, and the protein will be approximately 34. Okay. Now, in the 30s ribosome, the rRNA will be 16s, okay, and the protein will be approximately 21 amino acids will be there. This is how it will be, okay. So, in the larger subunit, 23s is also there, 5s is also there. In the 30s smaller subunit, 16s RNA will be there, okay. So, all these things are very important. We can expect in the question. Now, the eukaryotic ribosome. So in the eukaryotic ribosome, that is 80s. Okay, so in eukaryotic ribosome we have studied that 80s type of ribosome is seen in the eukaryotic ribosome. Okay, so this eukaryotic ribosome can be classified into two, which means two subunit together we are calling them as an 80s. So 60s and 40s subunit. Okay, 60s subunit is considered as a larger subunit and 40s subunit is considered as a smaller subunit. And in 60s subunit, the rRNA will be 28s, 5.8s and 5s. Proteins, that is approximately 49. This 40s subunit, which is considered as a smaller subunit, they are having an rRNA 18s and protein will be approximately 33. So this is how it will be. Okay, so all these subunits are very, very important. Each subunit has their own function. We are going to discuss about that. Okay, so this is what with respect to the prokaryotic ribosome and the eukaryotic ribosome, 70s and 80s. Now, functional sites on ribosomes. Ribosome have 3 tRNA binding sites. Just remember, this is the very important thing. Okay, see, ribosome, each ribosome have 3 tRNA binding sites. So, one ribosome has a capacity to bind with 3 tRNA because 3 tRNA binding sites will be present in the ribosome. Okay, and what are all the 3 sites? That is A site, P site, E site. A site is called as an amino acyl site, okay, where the entry of charged amino acids. Let me explain. Just know that A site is called as a amino acyl site, P site is called as a peptidyl site, and E site is called as a exit site. Okay. And what is the function of this A site? Where, see, if the tRNA attached with the amino acid, we can call them as a charged tRNA. If the tRNA is not having any amino acid, we are calling them as the uncharged tRNA. Okay, tRNA with amino acid is equal to charged tRNA. tRNA without amino acid is uncharged tRNA. Okay, now in the A site, that is in the amino acyl site, the entry of charged tRNA. So the first site where the tRNA has been entered for the first time, that site is called as an A site. Okay, that is what amino acyl site. And see here, this is P site is called as a hold peptidyl chain. Okay, P site is called as a peptidyl site, and in peptidyl site is the one holding the growing polypeptide chain. Let me explain with the picture also. And E site is called as an exit site, and where the exit of the uncharged tRNA. So if the charged DNA attaching site is called as an A site, and after adding one amino acid to the another, the growing polypeptide chain has been holded by this P site, P site, that is a peptidyl site, peptidide chain, polypeptide chain, and the uncharged DNA has been exit from the site, and that site is called as an E site. Okay. Now see here, this is an mRNA. Okay, so this is an mRNA. So in this mRNA, we are having smaller subunit and the larger subunit. Okay, so this is nothing but a ribosome. So as I have mentioned you before, each ribosome have 3 tRNA binding site. Okay, so in these 3 tRNA binding site, we are having A site, B site and E site. Okay, so in A site, where this is the tRNA, the tRNA with the amino acid is getting attached. This is the entry point, that is what an A site. Okay, after one by one tRNA with amino acid is adding in the A site. Now this has been shifted to the P site. This tRNA has been shifted to the P site. Now this P site has many amino acids. This is what the growing peptide chain. This growing peptide chain has been holded by this P site. Okay. Now the tRNA with no amino acid, that is what uncharged tRNA. This uncharged DNA has been removed at the E site. So this is what an mRNA. In the mRNA, this smaller subunit and the larger subunit has been bind. That is nothing but a ribosome. So in the ribosome, each ribosome, they are having 3 tRNA sites. That is A site, P site, E site. A site where the charged tRNA has been bind, and in the P site, the growing polypeptide chain has been holded by the P site, and the uncharged tRNA exit site is called as a E site. Okay, so this is very, very important. This is what the crucial function of the Yes. Now, ribosome biogenesis. How the ribosome is getting biosynthesized? See, you should know that ribosomes are important for the protein synthesis, isn't it? Now, you should know how the ribosome itself getting synthesized in the cell. Okay, ribosome biogenesis. Biogenesis in the sense formation. Okay. Now, in eukaryotes, in prokaryotes, how in eukaryotes they are getting biosynthesized, and in eukaryotes how it going to synthesize. See, in eukaryotes it is occurs in the nucleolus. Okay, nucleolus is the site, it is present inside the nucleus. So inside the nucleus there is another structure called as a nucleolus. Okay, so in the nucleolus there is an important site, that is important region, nucleolar organizer region. So in the nucleolar organizer region we can see the synthesis of ribosomes. Okay, so in eukaryotes the nucleolus will be there. In the nucleolus, the ribosome biogenesis will be taking place. So the rRNA genes transcribed by the RNA polymerase 1. So here comes another confusion. So we have studied in replication, that is when the DNA is replicated to another DNA. DNA polymerase is very important for that process. In the transcription, we are having RNA synthesis from the DNA. RNA is going to synthesize, and for this RNA synthesis, RNA polymerase is very important. And in RNA polymerase, we are are having three types of RNA polymerase. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Okay. So, and we have studied, we are having two types of ribosome. One is 70S and one other one is 80S ribosome. And also we are having subunits in both the types of ribosome. Each subunit have been synthesized by each type of RNA polymerase. That is, either it will be 1 or 2 or 3. See, RNA polymerase genes transcribed by the RNA polymerase 1. Okay, see here, rRNA. We are having three types of RNA. One is tRNA, and we are having rRNA, and we are having mRNA. And this tRNA has been synthesized by some RNA polymerase, and mRNA has been synthesized by RNA polymerase 2. And rRNA has been synthesized by RNA polymerase 1. And tRNA has been synthesized by RNA polymerase 3. Okay, and each subunit is also synthesized by different RNA polymerase. Just to see here, rRNA genes transcribed by RNA polymerase 1 except 5S rRNA. Okay, 5S rRNA is also the subtype of the rRNA. In the subtype of the rRNA, we can see the 5S. And except the 5S, all other subunits of rRNA has been transcribed by the RNA polymerase 1. Okay. Now this 5S rRNA, who is going to transcribe this 5S rRNA? That will be synthesized by the RNA polymerase 3. This can be expected in the question. Okay. Now this ribosomal protein synthesized in the cytoplasm imported in the nucleus. This is very important here. See, the proteins required, the proteins in the ribosome has been synthesized in the cytoplasm. After it has been synthesized in the cytoplasm, it has been imported into the nucleolus. Okay. So after that, the ribosome is going to synthesis. Okay. Okay, so this is important. And in prokaryotes, the ribosome synthesis will be taking place in the cytoplasm. rRNA transcribed as the single operon, not this much complex in case of prokaryotes. Okay, so eukaryotes is a complex organism. That is why complex process is taking place. Just note it down. The ribosome biogenesis will be taking place in nucleolus in case of eukaryotes, in case cytoplasm in case of prokaryotes. Okay, and next thing. Yes, see here, this is the ribosome and this is the cytoplasm. So this is cytoplasm, and here we can see the RNA polymerase 1. Okay, so this RNA polymerase 1 rDNA. Okay, from the rDNA, rRNA will be synthesized. That process is called as a transcription. So we are having 18S, 5.8S, 28S in the nucleolus. Okay, now all these has been processing. Okay, which means post-transcriptional modification. 18S, 5.8S and 28S. Now, this 40S and 60S is nothing but the ribosome. Okay, so that is the 80S ribosome will be synthesized as 60S ribosome and 40S ribosome. This is nothing but a subunit of the ribosome. Now, all these things are taking place inside the nucleus. Okay, and this is what a nucleus. And this is the nucleus. Inside the nucleus, we are having a structure called the nucleolus, and the space between the nucleolus and the nucleus is called as a nucleoplasm. Okay, so in the nucleoplasm, we are having RNA polymerase 2. And we are having an RNA polymerase 2 and polymerase 3. Okay, RNA polymerase 2, they are the one going to transfer into the mature ribosome. And this 5S has been transcribed. 5S has been synthesized by the RNA polymerase 3. Different enzymes involving in different function. Okay, now finally the mature ribosome, 80S complete ribosome has been formed in the cytoplasm only. Okay. Now, so we are, as we have mentioned before, we are having two types of ribosome. One is free ribosome and another one is membrane bound ribosome. Okay, so the free ribosome will be present in the cytosol, and the membrane bound ribosome has been present in the rough endoplasmic reticulum and in the nuclear membrane. In the membrane of the rough endoplasmic reticulum and in the membrane of the nucleus, we can see the ribosome. If it is present in the membrane, we are calling them as the membrane bound ribosome. And the protein synthesized and free ribosome, and where the protein will be synthesized in the cytosolic proteins, and here it will be secretory protein, lysosomal protein, membrane proteins. All these type of proteins will be synthesized by this membrane bound ribosome, and here the cytosolic proteins will be synthesized by this free ribosomes. Is there any structural difference between this free ribosome and the bound ribosome? No. Okay. There will be no structural difference. See, the difference is functional, not structural. Okay, as we have studied, we are having two different types of ribosome as free ribosome and membrane bound ribosome. Based on their structure, there will be no difference. Both the free and the membrane bound ribosomes are structurally similar but functionally different. So this is how mentioned here. See here, this is the membrane. If the membrane has the ribosome, if the ribosome is bounded with the membrane, we are calling them as the membrane bound ribosome. If it is a free ribosome, it is present in the cytosol, it will be looking like this. Okay. Now, the ribosome which is involving in the translation process. Okay, translation. So translation is nothing but the protein synthesis from the RNA. RNA has to synthesize the protein, and that process is called as a translation. And in the translation, we are having three important steps. One is initiation, elongation and termination. Okay. So in the initiation, all the ribosomal subunits has to be assembled on the mRNA. So as I have shown you in the picture, this is the mRNA, and we are having here a smaller subunit and here a larger subunit. In the mRNA, first the assembly of ribosome has to be taken place. The assembly means what? The smaller subunit and the larger subunit has to be assembled on the messenger RNA. So this is what the initiation. In the this is not with respect to the translation process. This is respect to the ribosome which is involving in the translation process. So in the initiation process, the ribosome is getting, the ribosomal subunits is getting assembled on the mRNA. And in the elongation process, peptide bond formation. Just now we have studied, we are having three sites. The ribosome have three tRNA binding sites. One is A site, P site and E site. Each site have their own function, and in the elongation, peptide bond is going to form. Okay, and in the termination, the release of completed polypeptide. This is happening in the E site and this is in the P site. Okay. Now, peptide bond formation. So here we have studied the peptide bond formation. So this peptide bond formation, how this bond is getting formed with the help of 20S rRNA in case of prokaryotes, 28S in case of eukaryotes. Okay, so this can be expected in the question. So which rRNA is involving in the peptide bond formation? And you should know 23S rRNA in case of prokaryotes, 28S rRNA in case of eukaryotes. Okay. Now what is meant by polyribosome? So I think we have studied this term before itself. Okay. Polyribosome and polysome. Polyribosome is also called as the polysome. Multiple ribosome translating a single mRNA. So there is a single mRNA. So we are having one ribosome, two ribosome and third ribosome. Many ribosome which is present on the same mRNA, and they are translating the protein simultaneously, and this is called as the one mRNA, many protein. This is what polysystronic. Okay, if one mRNA, many ribosomes, we are calling them as the polyribosomes, polysomes. Okay. Increase efficiency of the protein synthesis. The protein synthesis efficiency has been increased. Okay, found in both prokaryotes as well as the eukaryotes. Okay, the protein synthesis of this polyribosome will be present in both prokaryotes as well as the eukaryotes. And we are having the organelles which is present which has the ribosome within it. Okay, and we are having mitochondria and the chloroplast. The mitochondria is having the DNA and chloroplast is also having the DNA. And if there is a DNA, the ultimate goal of DNA is to synthesize a protein. For protein synthesis, definitely there will be the involvement of ribosome. So that is why this mitochondria and chloroplast have the ribosome within it. Okay, and what type of ribosome has been seen in mitochondria and the chloroplast? That will be the 70S ribosomes. Okay, so we are having 70S and 80S. 70s ribosome will be present in the cytoplasm, mitochondria, chloroplast, and in the prokaryotes. Okay, and in the cytoplasm of eukaryotes, we are having 80s type of ribosome. Now, similar to the bacterial ribosome, how the ribosome if present in the bacteria, the same structure will be seen in the mitochondria also, because both the type of ribosomes are 70s ribosome. And this will be the evidence of the endosymbiotic theory, and this endosymbiotic theory has been discussed clearly in the first class itself. Okay, so this is what this mitochondrial ribosome will be the evidence for the endosymbiotic theory. And the chloroplast ribosome, they are also having the 70s type of ribosome, and they are similar to the cyanobacteria. Okay, so if you know the endosymbiotic theory properly, if you have any confusion, go to the first video and see again the endosymbiotic theory and come here. Okay, now see here, antibiotics adding on the ribosomes. This is very important in an exam point of view. So antibiotics acting on the ribosome. How all these antibiotic target, which antibiotics is going to target which subunit of the ribosome. See here, so the streptomycin, tetracycline. So these are all the two types of antibiotic. They are targeting the 30S type of ribosome. Okay, 30S subunit. And the streptomycin effect will be misreading of the mRNA. If the message from the DNA is not transcribed properly, then the protein synthesis is also a problem. Okay, if the tRNA is not reading the mRNA properly, then again it is going to the protein synthesis is getting affected. Now the streptomycin is going to target the 30S subunit of the ribosome. It results in the misreading of the mRNA. Okay, and tetracycline again, this is also going to target the 30S ribosome, and here they are blocking the A site. If the A site is getting blocked, the charged tRNA has no binding site on the ribosome. Okay, so that is what, if there will be no charged tRNA bind, there will be no point of add, there will be no amino acid has been binding with the, no amino acid has been taken by that particular tRNA. Okay, so the A site is getting blocked here. Chloramphenicol is an antibiotic, and it is targeting the 50S ribosome, which is the larger subunit, and this inhibits the peptidyl transferase enzyme. If the peptidyl transferase enzyme has been inhibited, there will be no peptide bond formation. No peptide bond formation is going to take place. Okay, next, erythromycin. Erythromycin is an antibiotic. Again, it is also targeting the 50S ribosome, blocks the translocation. See, we are having A site, P site and E site. Okay, from the A site, the tRNA has to be translocated to P site, and from the P site, it has to translocate to the E site, and from the E site, the tRNA has to exit at that particular site. If the translocation has been blocked, there will be no protein synthesis will be taking place. Okay. Puromycin, very important antibiotic. So this puromycin targets both 30S as well as the 50S. Okay, premature chain termination. If the chain has been terminated before it has been matured properly, before all the amino acids has been added properly, before all the amino acids has been added properly, the chain has been terminated. So this puromycin has been targeting the premature chain termination. Antibiotics are selectively affect the prokaryotic ribosome. See here, the antibiotics selectively affect the prokaryotic ribosome. Okay, so this is very important statement. Now, what will be the difference between the ribosome and the lysosome? Okay, see here, the membrane is absent in the ribosome, and the lysosome, the membrane is present. It is a single membrane bound organelle. Okay, and the function of ribosome will be the protein synthesis, and the function of lysosome will be the intracellular digestion. We know that protein synthesis will be done by the ribosome and digestion will be done by the lysosome. rRNA plus protein, we are calling them as an enzyme. A group of enzymes, we are calling them as the lysosome. Okay, and we have discussed the lysosome top to bottom in the previous class itself. Okay, now So these are all the important key points with respect to an exam. See, ribosomes are non-membranous bound organelles, very important. Okay, and rRNA is catalytic. So the enzyme present in the ribosomes is ribosine. Okay, so very important. You can expect this in the question. And see here, all the subunit has been transcribed by RNA polymerase 2, but here 5S RNA has been synthesized by the RNA polymerase 3. Very important. This can also be expected. See, ribosome attached to the RER. Why it has to be attached with the endoplasmic reticulum? To synthesize the secretory protein. No. Endosymbiotic theory, and in endosymbiotic theory the organelles will be mitochondria and the chloroplast, and the type of ribosome seen in these two organelles will be the 70S ribosome, no doubt in that. Okay. And in the exam, how can the questions can twist us? See, S value is not additive. So we have mentioned as 5S plus 30S. Okay, and we have said that the 70S ribosome has two subunits. One is 50S and the 30S. 50S and 30S, and together if we add both these things, if we has 80S, but here only 70S. So that is why they are saying that S value is not additive. It is nothing but a Svedberg unit. It is a sedimentation constant. Okay. Now, free and bound ribosome are structurally identical. So you should not think that, as I have mentioned you before, the ribosome is present as free ribosome in ribosome, a free ribosome in cytosol, and it also membrane bound. Both are structurally similar, but their function is different. Okay. Ribosome is again, it is absent in the mature RBC. This is very, very important. So this is a one-line quick revision. So ribosome is a protein factory, we know that, and the subunits of 70S and 80S will be present in the prokaryotes and the eukaryotes, and we are having three tRNA binding sites in the ribosome, A site, P site and E site. rRNA is catalytic RNA, we can call them as ribosine. Okay, and nucleolus is the ribosome factory. Okay, nucleolus is the ribosome factory means what? Ribosome biogenesis will be taking place in the nucleolus. Okay, so that's all about the ribosome. A complete information has been taught now. So what is mean by ribosome? I mean, now comes to the question. Just see here, the ribosomes are classified as ribonucleoprotein particles because they contain. Now, what do they have? What do they have? Just pause the video and answer the question. So, we have studied RNA plus protein is called as the ribosome, and the answer for this question is option C. Okay, rRNA plus protein, hence nucleoprotein complex. So this is what a proper answer for this question. Now, which rRNA molecule plays a catalytic role in a peptide bond formation in prokaryotes? So, this is very important here. So all these are considered as, all these were the subunit of rRNA, we know that. And which exactly involving in the catalytic role in peptide bond formation in prokaryotes. Okay. So, in prokaryotes that is 23S rRNA, in eukaryotes it will be 28S rRNA. Okay. Now here the statement Svedberg units are additive is. Svedberg unit, that is yes. Yes units are additive or not additive. Whether it will be true or false. Just now we have studied it is a false statement. Okay. Because sedimentation depends on shape and mass. Okay, it is not additive. So the answer will be option B. Okay, and which of the following ribosomal RNA is synthesized by RNA polymerase 3? So RNA polymerase 3, it is synthesizing the 5S rRNA. We keep on studying the same thing. Okay, don't forget all these things. Free ribosome and RER bound ribosome differ in their structure, size, function of protein synthesized, rRNA composition. Structurally, they are similar. Okay, their functions is different. Okay, differ in the function of protein synthesized. Which site of the ribosome is occupied by the initiated tRNA during the initiation of the translation? That will be the P site. Okay, so what do we have studied? So we are having E site, P site and A site. The charged DNA will always be bind with the A site, but the first tRNA will come and bind with the P site. So this process will be clearly seen in the molecular biology chapter. Don't worry about this. But as of now, just know we are having 3 sites in the ribosome, that is E site, P site and A site. So the tRNA has to be bind here, the charged tRNA, but the first tRNA will bind with the P site. Okay. Now, the presence of 70S ribosome in mitochondria supports, that will be the endosymbiotic theory. Okay, and which antibiotic inhibits the peptide bond formation by acting on the ribosome? Which antibiotic inhibit the peptide bond formation? And that will be the chloramphenicol, no doubt in that. Next, polyribosome, polysomes. This indicates our multiple ribosome translating one mRNA. Okay, so this is the correct answer. And ribosomes are absent in the mature mammalian RBCs. Okay, just now we have studied. Okay, so in this class we have completely discussed about the ribosome. We'll meet you in the next class with some other organelle. Thank you so much.